coagulation factors. How many factors are there? 13. You write in your copy. 1, 2, 3, Roman 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. Some words. You have to write sequentially the initial letter of that word. Okay? Farid. Farakka. Faridabad. So, initial letter is F. So, write F. Then you write Faridpur. Pur means P. You have to write P. Faridpur. Training. T. Training. So you have to write T. Farid, then Pur. P-E. Yes, right. T. Training. College. Faridpur Training College Labor Association साहब को इतना पीटा, ऐसा पीटा, सांस प्रोसेस হয়েছে লোপ, lost. So this is the thing you have to remember. What? Faridpur Training College Labor Association साहब को ऐसा पीटा, A, P, सांस process, this is process, inhalation. Faridpur Training College Labor Association साहब के नो के साहब नो के एक बार ऐसा पीटा, तो सांस गया, प्रोसेस भी गया, मीनिंग इनहेलेशन एक्सहेलेशन गन, ऐसा पीटा. देन सांस प्रश्वास हुआ. H लोप का हिंदी में क्या मतलब? लोप, lost. वो तो नहीं आएगा. L, you have to write L. So lost. सांस process lost. Okay. Now write F for fibrinogen. F means fibrinogen. Now you got the sequence. Then prothrombin. And T for tissue thromboplastin required for extrinsic path. This will initiate the extrinsic path. So, what is this? Tissue thromboplastin. And next is C for calcium. Very simple. Calcium ion. And L for labile factor. Labile factor. A is absent. Previously its name was accellerin, but its existence not proved. So it is absent. Then what is there? Stable factor. S for stable factor. And A for anti-hemophilic factor. A for anti-hemophilic factor. So now you can see, in absence of this factor, there will be hemophilia. Since the name is given anti-hemophilic factor. So this one is factor 8. 8, no? It will cause hemophilia B. What type of hemophilia? Hemophilia. Remember this. Absence of this protein will cause hemophilia B. And then plasma thromboplastin antecedent. Plasma thromboplastin antecedent. And factor 10 is, we know, Stewart factor. Stewart-Prower factor. Stewart-Prower factor. P-R-O-W-E-R. So, Stewart-Prower factor. Understood? Okay. Then, again, plasma thromboplastin. This is Stewart-Prower. Stewart. P-R-O-W-E-R. Prower. W, P-R-O-W. Cut it, yes. Yes, okay. The power factor. And the 11 one is P, plasma thromboplastin. First one is not written, component. Thromboplastin component, right component. Yeah, component. And that one is antecedent. Antecedent. Plasma thromboplastin antecedent, PTA.
Sir, that is component.
Huh?
Component.
একটু ভুল বললাম the plasma thermoplastin component first one is antecedent.
The 9 one is antecedent. Number
9 not component, antecedent. This is component. এটা antecedent. Antecedent. This is antecedent. This is component. Sedent. Antecedent. And next one, Hagman factor.
Yesterday you showed that 11 is antecedent and 9 is component.
Okay, let's see. First one is component. 9th is component. Oh, it was correct. Hey, you misguided me.
component was
You misguided me. So I lost confidence. So initially I was right. And this one is Lucky Laurent factor. Okay? Done? So what is this? Faridpur, not a single word, separate word. Faridpur Training College, Labor Association, Saipko, Ezapita, SAS process, who are lost. So now you can remember the names. You will not forget. Now you write antecedent. ঠিক আছে আর ওটা antecedent and earlier one is component. No, that is correct. That is correct. Initial one is component, later one is antecedent. Okay. Now we have to study the mechanism. It has got 3 steps. Mechanism of what? Coagulation. So it happens in 3 stages. What is the initial stage? Formation of prothrombin activator. Formation of prothrombin activator in which thrombin will be activated into prothrombin. Prothrombin will be activated into thrombin. Prothrombin activator. This is the enzyme who will convert prothrombin into thrombin. So up to the formation of the enzyme, this is the phase 1. Next phase is formation of thrombin, prothrombin to thrombin. And third phase is fibrinogen to fibrin. So once fibrin is formed, then coagulation is over. So these are the three phases. Let us remember what are the factors will be involved in. Now it will be very easy for you. Involved in intrinsic factor. Right, factor 12. Right. Factors for intrinsic path. Then factor 9 will be coming. Sorry, factor 11 will be coming. Then factor 9 will be coming. 9 means 12 is required, 11 is required, 9 is required, 10 is required. So actually 10, 11, 12. 9, 10, 11, 12. These are required. But I said sequentially. 12, reduce 1, 11, then 9, again you increase it 10. So these are the factors will be involved in intrinsic path. How they will be involved, that I will say. And in extrinsic path, 2 factors. 7 and 10. 7 and 10. So what? Now I can remember. I think everybody has written. Where?
What?
You have to write what are the factors. What I am telling. Where we had been. No. That I am telling you to write. I am helping you to learn. Not disturbing you.
Okay, sir.
I am helping you. Try to remember this. Okay. And what are the factors to be involved in for extrinsic path? Remember, both this path will produce one thing. What is this? Activator. Prothrombin activator. That is the function of the both path. And after that it is common. After this there is common. Later we will find there are some tests which can identify there is problem in either there is problem in the intrinsic path or in the extrinsic path. There are some tests. So with that, these are the significance of these tests. After that, what are the blood coagulation or blood clotting tests? So it is coming later part. So now we have got what are the factors involved in intrinsic path and in the extrinsic path. Okay. So intrinsic path, let us first start with intrinsic. Intrinsic path, can you follow? Okay. Intrinsic path, what is the first factor? So it will initiate its reactions. For initiation, it will look forward for one thing. What is this? Collagen. Where it will get collagen? Because there in the ruptured wall, there is collagen, exposed collagen wall. So when the factor 12 will come in contact with this, immediately it will be converted into factor 12A. It will be activated from inactive form to active form. Is that okay? So you write. So in place of enzyme, here you write collagen attachment. Not attachment, collagen touch. Collagen touch will activate factor 12 into factor 12A. So in this reaction, some local hormones will be involved. You just write at the side. HMG, kininogen. It is there, no? HMG-C, kallikrein and HMG kininogen. Okay. Now next phase you see, first phase done. Factor inactive 12 has been converted into this. How? This is initiated by collagen. Initiated by touch. When definitely while passing these plasma factors, while passing through the damaged area, so it will come in contact with the collagen and immediately it will be activated. This is an active enzyme. All these factors are other than two. Two are not enzymes. One is tissue thromboplastin and another is calcium. All other are actually proenzymes. When they are activated, they form enzymes, active enzymes, and they will do some reactions. Okay. So in the next phase, what will be what is the next phase? 11 will come. 11 will be activated by 12A, and what will be formed? Active 11 means 11A. So write the reaction. Yesterday, why I am giving emphasis on this? Yesterday, I have gone through several university exams of different universities. This is one of the commonest question. Describe the intrinsic path or the extrinsic path. So it is a frequent, this question comes frequently. You must learn this. You don't know whether during your exam it may come. It is a long type question. So this is formed. What is this? This is formed. This is transformed into this, and now factor 9 next will take part 9. So 9 will be converted into 9A by the previous enzyme. Okay. So here it will require presence of calcium ion. We take out our blood. If we don't want the blood to be coagulated, then we add some anticoagulant. Okay. What they do? So, for example, oxalate. This oxalate will remove the calcium from the plasma. So all calcium will go with oxalate as calcium oxalate. So there will not be any calcium ion left in the medium to participate in the reaction. So there will not be coagulation. The blood will not be coagulated. So this is the mechanism. So next what will happen? Now this will be produced. How? This enzyme will convert inactive 10 into active 10A. What is the name of this inactive one? Stewart factor. So now when it is activated, we don't say it is Stewart factor A, we call it prothrombin activator. 10A means prothrombin activator. So our intrinsic pathway is over. Intrinsic pathway is up to this. Same product, same 10A will come from extrinsic path. Then these both products, same product, will participate in the next phases. Okay? So if intrinsic pathway comes, to initial part you have to explain. Up to this you have to explain. Then you have to explain the phase 2 and phase 3. Then coagulation will be completed by this process. Next 2 phases are common to both paths. I hope you will be able to remember this. Later part is common. So origin of intrinsic path is what? Who stimulated this path? Collagen. Exposed collagen. But what about? That's why This, see, collagen is just its presence, but not participating in any reaction. That's why it is called. Though all these factors are the product of plasma. That's why we call this. That's why it is called intrinsic within the blood. That is the reason we call it intrinsic. There is no participation of any component from the tissues or other than the tissues. Okay, their only thing is their presence of collagen, only just touching, no direct participation in the reaction. That's why it is intrinsic. Can you remember this? Will you be able to remember this, Shardaji? Okay. So next, we'll discuss the extrinsic part. What are we already told? So here let's see who initiates the reaction and it will participate in the reaction. What is this? The tissues are having so many things other than collagen. The tissues will be having some internal within the cell. There are some components, phospholipids, lipoproteins. So these phosphoprotein, lipoproteins are actually tissue thromboplastin. And see, this is participating in the reaction. See, this is participating in the reaction. This is factor 3. That's why it is extrinsic, not intrinsic. Started with extrinsic part. What is the source? Again, you try to remember. From where this factor 3 came? There are so many types of tissues now on the wall. Tunica adventitia, there are some cells, connective tissue cells. Okay, in the muscle, there are muscle fibers, and in the intima, there are epithelials or endothelial cells. Endothelial cells also secrete something. They also participate in the reaction. So, the tissues are liberating some products, their own products. We call them extrinsic, extrinsic, with this one, tissue thromboplastin. These are actually what? As I said, lipoprotein, glycolipid, these are the products. Some proteins are also there. Okay. So, when the tissue is traumatized or injured, then only this component will be coming out from the cell. Then only this process will be initiated. Okay. So, at the first phase, what happens? This tissue thromboplastin converts factor 7 into 7A. This path is very simple. Then factor 7 will convert as enzyme the inactive Stewart factor 10. It is just 10 means Stewart power factor and active 10, that is prothrombin activator. So extrinsic path is complete. It is completed, and now this both path has produced. This is prothrombin activator. This is also prothrombin activator. So they join together. They are coming together. Now they are concentrated much more. Every activator can stimulate, convert near about 100 prothrombin into thrombin. So here the reactions will be very fast. Okay, enzyme activity is very fast. Now see, this is the intrinsic pathway for the formation of 10A, and this is the extrinsic pathway for the formation of 10A. So this 10A is coming from this source and also from this source. Now they will follow the common path. So the second phase is this. Second phase is this. This 10A will convert prothrombin into thrombin. Prothrombin, these are made by liver. So if there is any liver disease, what will happen? Fibrinogen will not be produced. Prothrombin will not be produced. There are some other factors will not be produced. So there will be coagulation disorder because of the damage of the liver. In the third phase, fibrinogen will be, fibrinogen is a pro-enzyme, not enzyme, this is not an enzyme, it is a protein. This is not an enzyme, this is a protein. Proteins are also, enzymes are also protein, but all proteins are not enzymes. Okay, so this is not enzyme. These are having polypeptides. So now by the action of the thrombin, this will be splitted. So there will be numbers of monomers. These monomers will join. So from these monomers, polymer will be formed. Fibrin polymer will be, this will be done by the factor 13. Now factor 13 is involved. Polymerization is done by factor 13. Not only polymerization, hardness means insolubility will be rendered by factor 13. When it is made insoluble, now this fiber is ready. So now the function is to make a net. So it is now formed a net. And formation of this net is actually clot. And because it forms a net, clotting is done. Because it is the property of the plasma, not the property of the RBC. So now since the net is formed, now there will be trapping of cells. And all these activities, where it is going, where it is undergoing, only within the injured area, cut area, not anywhere else. If it is continuous anywhere else, within the blood vessel, there will be clot. So this blood vessel will go. No blood will be there. So I will not allow, because my inner wall is having some preventive mechanism. What? I will not allow the rest part, lumen, will not allow any coagulation. If any chances there, immediately it will come into action. When this will be overpowered by some other factors, then there will be coagulation, formation of thrombus, and this thrombus may create lot of problems. It may cause heart attack. It may cause brain attack, means stroke, by blocking the blood vessels. It may cause pulmonary embolism. Okay, it may, see, DVT and another disease, leg veins. We know veins have some valves, no? So that made them flow of blood unidirectional. Only from leg it will go up. It cannot go back because some valves are there. When it is going out, it cannot come back because of the valves. So when your leg, what you are doing? For long period, you are sitting like that, or some obstruction is there. So there will be lack of movement of blood in the extremities. And what it will cause, you know? The blood is still, no, now it can't flow. So there will be clot formation. There will be clot formation. It happens in patients who are lying in ICU bed, who are in coma, lack of circulation. So there will be formation of clot within the. So what we do? So that clot is not formed, we take precautionary measures. What? precautionary measures? We will try to prevent coagulation by adding coagulant. What type of? There are some therapeutic use of coagulant like heparin. There are some other oral anticoagulants, IV anticoagulants. The patients who are suffering from heart attack, stroke, obviously they will be given, 100% they will be given, and these anticoagulants. There is question comes. What is the mechanism of action of heparin? Write down. Mechanism of action of heparin. How it prevents coagulation. Docoumarin. DOCOU. MARIN, docoumarin. What is their mode of action? They also prevent, either they will prevent coagulation, or if already blood is clotted, they will help in breaking down this thrombus into pieces, so that these pieces can be engulfed by the phagocytosed by the macrophages. So, lot of macrophages, they are wandering within our tissues, within our blood, saving us from various disorders. Very helpful. And where they are produced? In the bone marrow. They are present in liver. They are present in intestine. Everywhere they are present. They are wandering tissue, wandering everywhere. Gurivera. So I hope all of you now are able to describe intrinsic path, extrinsic path. You will not be asking me, sir, please describe this path. Yesterday, why I am repeating this? Because some student asked me to again explain this. Then I realized the other students may have same problems. That's why I took this class, repeated this class. I think this was helpful to you? Okay? Fine. No. It is coagulation cascade. This is a video you saw. So no need. And clot retraction, 2 marks, right? You have to study clot retraction. I will not repeat. How these are retracted? Clot, and the RBCs, platelets, they are connected by the fibrin. So plasmin will cut this thread. These are enzymes. So what will happen? If you cut a net where there are a lot of fishes, so if you cut the thread, these fishes will be coming out, they will be freed. So here also, these small pieces will come out fragmented. This is called fibrinolysis. Lysis of the fibrin, not lysis of the RBCs. The lysis of the fibrin. Okay? See, marked, highlighted part, what is written? Important. Why it is happening? What is the need of it? Need is this. Because I have to repair the injured area. So, but this area is occupying by the clot. So, okay, bhai, you auto yahan se, let me come, let me join. Let us join the both ends together. So that's why they are this will be eliminated from this through the process of fibrinolysis. lysis. That is the importance. And so wound healing and prevent fibrinolysis. Okay. If it is immediately after formation, is why it is retracted? So that, see, there are initially platelet plaque formation that was weak, that was temporary, but this is stable. This clot is stable. Now blood cannot pass through. So it will not allow, our blood will not allow to stay longer, because at the same time I started the process of repairing. So for that purpose you have to come out from there. So how it has been done? By fibrinolysis. So clot retraction, fibrinolysis, both are important. Short question may come. Or write short notes on this. So this is the mechanism of fibrinolysis. So you have to write this, if possible, get a snap of this. My lecture is a mixture. Whatever I am showing, not some, so that I make it easily edible to you. So that for this purpose, I take some good portion from here, good portion from there. So ultimately, I made it. So everything you may not get in your books. But for your understanding, these are required. At this point, you have to explain 2 marks, 3 marks. Why blood does not clot in circulation. Get a photograph of this. And then explain. I will ask tomorrow why blood doesn't come. Not only heparin, there are some other factors. You have to remember those factors. Fibrinolysis is an anticoagulant mechanism also, because there is no injury. There is no injury. What I am doing, tying my hand here and restricting the movement. No injury there, but it will cause injury within the endothelial membrane. Since endothelial cells are also not getting their oxygen necessary thing, so it will also produce functioning abnormally. So it will start coagulation, and thrombus may be formed, and I may die. So our body prevents it. So immediately it produces some anticoagulant factors. Anticoagulant. Because of this, right? Protein C. Protein C, it negates. Whenever there will be any process, so it will inhibit. So initiation is restricted by protein C. There are some other factors. You try get a photo of this. Protein C and TPA. TPA stands for tissue thromboplasmin activator. When this will be activated, plasminogen will be activated into plasmin. So we know the function of plasmin. If there is any formation of clots, so immediately it will remove those clots. So these are the preventive mechanism why blood doesn't come. If there is any production or formation of clot, this will be destroyed. It is as a precaution. But sometimes we fail. When we fail, these preventing mechanisms are overcome by those. We are adding some drugs, taking some drugs, okay, or lying for long, in bed for long. So this may cause initiation of clotting, and our preventive mechanism may not be able to resist this. This is important. You will get this in your book. It is GK Pal. Some chapters are GKPAL are unique. And occasion just briefly mentioned, or sometimes it is absent there. Okay, but GKPAL, something, or there, another book is there. I don't know how many of you have purchased. That is other than guidance. What is this? Not Swaminathan. Shambulingam. Shambulingam. Yeah, that book is also good.
So GKPAL or AKJ, which is better? GKPAL or AKJ, which is better?
I told. Hello. There are some, it's like notebooks. But if you want more marks, then you have to add more points.
Yes.
So that you will get from GKPAL.
GKPAL.
Some part is better in GKPAL, not all. Sometimes it is too much is given. It is tough to assimilate. So everything is important, but everything is not important. Extra things are there. But in occasion, it is brief. So for first you get the brief knowledge. So go through the occasion. Then you, as reference, you study the other books. This is the tips. While studying a chapter, say for hematology, so for hematology there are certain units. So in the book it is also written like that. So at the end of each chapter, you will find some tips. For example, long type, from this chapter, what are the long type of questions may come? So these people who the author, they have studied different questions of different years, of different universities. Then they suggested this can be a long type question, this can be asked in Viva, this can be a short question, it can come as an MCQ. So these tips are given at the end of each chapter. So you go through these chapters, and possibly from the next week, every week we will be giving you certain MCQ online. Your email ID is there with the authority. Through this email, there will be every week you will be getting some MCQ question. Google questions are there. From there you have to just click the options, and immediately your result will come to you, and we will also get the result. It may not come to you, but we will get the result and we will say what is your performance. So, is it okay for you? Now you are not studying. You are not studying. I don't know what you are doing after going back to your home. I have doubts. So, if you have to be pressurized, for study. So this is one of the weapons. And then another thing will be implemented. We will be giving you short assignments. That too based on important questions. So you have to prepare this and submit it to the department. That will be written. For 2 months, 4 months, it is definitely will be helping you, and you will face lot of problems. Then you will be asking us, sir, this is the problem. What would be the correct answer? So we can assist you. क्या सरदार जी, क्या बहन ये ठीक है या ना? Okay. So there are many types of bleeding disorders. Bleeding disorders means abnormal clotting mechanism. Means either there may be delayed clot formation. So by this time lot of blood will be coming out. You know some diseases like that. Somebody is having patches and it looks purple in color. What is this called? Purpura. So it is a bleeding disorder. And some are having hematoma, swelling and because of liberation of some bloods and they are accumulated. These are called hematoma. So if there is any bruise, nobody hit you, hurt you, automatically it is developed in your body. That means something wrong with the clotting process and there is clotting disorder. So we'll study these clotting disorders and I will suggest you two clotting disorders you must learn properly and the mechanism. One is purpura, as I said, another is hemophilia, another is Christmas disease. Both are symptoms are same, only the factors are different, but the symptoms are same.
Third one is Christmas hemophilia.
Christmas disease. Christmas factor is there because of the absence of this factor. Huh?
After 9 Christmas factor.
Yes, yes, factor 9. See, there may be automatic spontaneous bleeding in the skin or below the skin, and it may be because of disease of the platelets or the factors or the disease of the wall, capillary wall, abnormality of the capillary wall. That's why see basal wall disorders. So from basal wall disorder there may be bleeding. These are the various disorders. These are not important for your exams, but purpura comes under this. It is a basal disorder. And the platelet disorder, that means the platelets are abnormal, either absent or some abnormality is there. We have studied platelets are having some binding sites. If there is no binding site, it cannot bind to the collagen. So there will not be formation of platelet plug. So blood will be continuously coming out. There will be delayed clotting mechanism. So these are the defects. Or there may be some clotting factors, absence of clotting factors like factor 30, factor 10, factor 8. So they may cause problems. So there are some diseases that are involved in good. Some factors, some diseases will be there because of formation of or defect in formation of platelet plug. And there may be some defects during clotting mechanism. So both type of, one is the first one, hemophilia patient, bleeding in joints, swelling of joints because of bleeding, internal bleeding. So this is purpura, this is important, and this disease is also important. So because of lack of binding site on the membrane of platelets. This is the cause. You have to mention the cause. So you have to know about this disease. Thrombosis, thrombus, formation of no injury. Blood, there is formation of clot inside the blood vessel, within the lumen of the blood vessel. Why it happens? Injury of the endothelium by infection or by mechanical, as I said, obstructing the and by a blood pressure cuff and roughened endometrium lining, as it happens in case of atherosclerosis, deposition of lipid in the innermost layer. endothelial cells. Sluggish flow. While lying for long period on bed, paralytic patients, they may suffer this. Agglutination of RBC, mismatched blood transfusion. This may also cause formation of clot thrombus, toxic thrombosis, snake bite. I am also a snake. Nag means, so that's why be alert. Anytime I can bite you. Toxic thrombosis. Which snake bite is most toxic? Russell Viper. No chance. One bite. Finish. No time. না ছোট মাথাগুলো একটু মোটা টাইপের রাসেল না রেটল স্নেক তো রেটলিং করে নয়েজ করে রেটলিং ইট রেটলস and hello particularly 2 or 3 anticoagulants come in your exams. Write it down so you have to prepare it. I will not take any class on that. One is heparin, anticoagulants. When we give anticoagulants, either in stroke patient or in heart attack patient or in pulmonary embolism or to prevent these diseases. Some are having chances. No, when my blood is tested for lipid profile, it is found that HDL cholesterol is much less. LDL cholesterol is high, triglyceride is high, free cholesterol is high. So there is a chance of formation of atherosclerosis. So there is a chance of blockage of blood vessels, means it may happen in heart or in brain. So there is chances of stroke and or heart attack. So as a preventive mechanism, I will give anticoagulant. So in such case, those who have already suffered an attack, they will be given this anticoagulants. So you have to know the mechanism action of heparin and mechanism of action of this warfarin. This is vitamin K antagonist.